Yeah, so I will quickly go through how you can make your modeling life simpler. And the goal is that you are not sticking to a single tool. So we teach here Copasi and Virtual Cell, but there are other tools like Cell Designer, Tellurium, and so on. And the major format that these tools talk between each other is through SBML, Systems Biology Markup Language. When you go to File menu on each of our tools, you will see Import Export, and one of the options would be SBML. You may be confused that there are different levels of SBML that are supported by each of the tool. So basically, you just open two tools, you see which is common. Usually level 3 is what is supported by all tools. So when you do this import-export, you have to be careful. Sometimes you will get warnings, sometimes you even may get errors, and still, even with errors, you will be able to go ahead and import SBML from one modeling tool to another. So what you have to be aware of: names, units, and values. So you successfully imported model that you exported as SBML from one tool into another tool. Don't be very relaxed. Go through the names and IDs. And this is important. In Copasi, there is even an option like strip SBML IDs. And in Virtual Cell, we display separately Virtual Cell or ID and name. The reason is that ID are unique, but sometimes they are called absolutely in a stupid way, like species underscore 1, 2, 3, 4, 5. And names are readable, but they may have empty spaces not supported by one or other tool. They may have repeated names and so on. So each tool deals with them separately. So if you will import from one tool into another one and see some strange names, don't worry. Just try to match them or try to kind of open SBML file and see what's going on. Units, sometimes different tools handle them differently, and values. So it's rare, but you have to be aware of it. Another format that is used in Virtual Cell for rule-based model, it's BNGL. And again, there is a bioenergen.org, which is a standalone tool, and it has no graphical user interface, but it has an editor, and it has some features that are not supported by Virtual Cell. So you may want to move files from bioenergen to Virtual Cell. Virtual Cell supports compartmental modeling and compartmental NFC, which is not supported by standalone bioenergen. So you may benefit from either of the two tools. And when you export import through BNGL, you again may have some issues. And these issues, they may look scary, but they can be easily fixed by just in a text editor. And Virtual Cell helps with that. When you import from bioenergen to Virtual Cell, it opens an editor and it points out the lines that have to be fixed. So generally, there are formats that are modeling: SBML, VCML, Virtual Cell, Copasi, bioenergen languages. And there are three other categories of formats that you may encounter. It's biopacks for databases, SBGen for visualization, and ZML for simulations. And ZML is something that both Virtual Cell and Copasi support, and some other tools support it as well. So yeah, we'll talk about it in a second. But about modeling resources, we have a lot of different external resources that can be used to get models in SBML format. So it's biomodels database, Reactome, basically almost any pathway database now supports export in SBML format. And again, there are some warnings that will be raised. Say some compartments may be unspecified, some names can be strange, but you can use many pathway databases to extract SBML format. SBRK is a reaction kinetics, and then just any SBML model that is driven is either designed in other tool or just journal publications. They have supplemental material if they have a model, and very often this supplemental material has SBML format. And then finally, SDML is something that you will see in both import-export in Copasi and Virtual Cell. This stands for Simulation Experiment Description. And if you use Virtual Cell, you know that Virtual Cell is like a tree. When it's not just a model, but you also can have an application that tell you how this model is customized. And then you have simulations when it's written for how long you simulate model, which simulator you use, and then which, no, time course is not, but just simulator and time. Note that SBML doesn't store this information. SBML stores just the model itself. So this additional information is stored in SDML, and it's not very useful for exchange of information between Copasi and Virtual Cell, but it is very useful for a new tool which is called biosimulations.org or biosimulators.org. And in these web tools that are now under development and recently released, you can load SDML files from both Virtual Cell and Copasi and run them varying parameters. The advantage, you can do it online in the web browser without loading and working with Copasi and Virtual Cell as the desktop applications.